Hi everyone, I hope you are all doing well. So in today's lecture we are going to be looking at angiosperm germination. And really this is the start of a series of lectures that are going to focus on primarily on plant anatomy. And that's going to take us through really the first third of our course all the way up to your first test. So before we dive into this, let's review last lecture. All right, so in last lecture, we looked at ecosystems and agriculture. And we started by saying kind of two main points. The first is that a crop system is an ecosystem. And then the second was that the more a crop system varies from the natural ecosystem found in an area, the more inputs and resources are needed to grow and sustain crops. And that was kind of the thesis of the entire lecture. And from there, we went on to actually talk about what an ecosystem is, right? Remember, it is a community of living organisms and the abiotic or non-living environment where they're found. And we said that ecosystems have four main components: geologic and soil features. And then when we were talking about plants, why isn't that working? There we go. And when we were, and it's red. When we were talking about plants and what plants are going to grow in an area, we said what was important was the relief of the land, right? So that's essentially the landscape. Are there mountains, valleys, etc., slopes, and the soil. And there are a couple things important with soil. The texture, because that's going to determine the water holding capacity of the soil. And the other is really the percent of organic matter. And in a lot of agriculture soils, you want it to be between 3 and 6%. That's kind of like a good percentage of organic matter. And we went into a little bit more detail with that. Then we said besides geologic and soil features, we had climate. And within our climate, we had what was important for plants were temperature. Whoops. So I'm having all sorts of trouble here with these today. What the heck? Go back to normal. Oh. What is going on? Right. Maybe I can actually get through this. So for climate, it was temperature was important. Holy Lord, I'm losing my mind. Temperature. All right, temperature was important. That's going to dictate how fast our plants grow. We had precipitation. Precipitation. Right? Water is important for photosynthesis and some other processes that we'll learn. And then down here, temperature, precipitation, and light. Right? The amount of light received in an area. And again, light is important for photosynthesis as well as some things that we will learn later on, such as photoperiodism or photomorphogenesis. So that was climate. Then we had plants, animals, and other biota, and we said that they could have both positive and negative impacts. And we had humans, which were technically animals, but because our impacts are so vast, we get our own separate category. So those four categories make up an ecosystem, and they can all impact what plants will naturally grow in an area, as well as what would be good crop plants to grow in an area. And kind of talking about that, we brought up this concept of sustainability. And that is the ability to be maintained at a given level. When we were talking about the environment, we're typically talking about maintaining environmental resources or striking an ecological balance. And we said when we are talking about a crop system, one way to improve sustainability is to mimic nature. And we can do this in a couple of ways. We can mimic the ecosystem characteristics, right? So if we look at natural ecosystems, what are some of their characteristics? They're often diverse. There's a whole range of different plants that are growing at different stratum. So you have trees in the canopy, and you have a subcanopy layer, and then your herbaceous layer. So there's diversity, and with that diversity of trees, you get diversity of insects and animals that can help pollinate, protect against other bad pests, etc. So we can mimic some of these ecosystem characteristics. We want to match our crops to the natural habitat, right? So we should know what are the habitat characteristics in an area, what will grow there naturally, and then we want to match really what we're going to be growing to what works well in that area, right? And the example I gave was blueberries in Hamilton, right? Blueberries are a heath plant. Heath plants grow well in sandy acidic soil, which just so happens to be the soil that you will find in Hamilton and really throughout the Pine Barrens. And then we can also utilize ecosystem services. This kind of goes hand in hand with the first one, right? Ecosystem services are just services that different aspects of the ecosystem can provide to us. I talked about the mycorrhizal fungi that in wild plants, it forms an association with the roots and it helps extract nutrients from the soil. We can incorporate that mycorrhizal fungi in our crop systems. We do it in our greenhouse. We're just growing our plants in there. The video I had you guys watch looked at fish and incorporating fish and ducks into a rice paddy. And the fish acted to eat pests, and their feces fertilized the plants, helped fertilize the plants, whereas the ducks were really effective at weeding out the rice field. And then in the end, you're able to sell all of it, right? You can harvest your rice as well as your ducks and your fish. So again, a couple ways that we might want to mimic nature. There's plenty of examples. If this is something that is interest you, this is really, I believe, the wave of the future is being more sustainable in our agriculture. And this way, mimicking nature, I think, is one really good way to do that. So that's our review. Let's get on with the lecture. Okay, so if you are looking at plants as a whole, they can be divided into two main groups. And those are our seed-bearing plants versus our spore-producing plants. Right? We have plants that produce seeds, and we have plants that produce spores. Now, our seed-bearing plants are the plants that we're going to focus on. They include gymnosperms and angiosperms, which we'll look at in a second. Whereas our spore-producing plants are those such as ferns, mosses, liverworts, and green algae. We're not really going to focus on them too much. We're going to focus our time really throughout this course on our seed-bearing plants. Now, if we look at our seed-bearing plants, as I said, we can break them down further into gymnosperms and angiosperms. The main difference, gymnosperms are flowerless. They do not produce flowers. Angiosperms are flowering plants. That's the main difference. That's what I want you guys to know. Gymnosperms, they get their name. Gymno means naked, and that is because their seeds aren't enclosed in an ovary or fruit like our angiosperms are. But really, what you should remember is that gymnosperms are flowerless plants. Angiosperms produce flowers. Now, our gymnosperms are the older of the two. They evolved first. They evolved around 390 million years ago. And they were very, very numerous throughout the Mesozoic. So it's the time of the dinosaurs, right? When dinosaurs were roaming the earth, they were in these giant forests that had these large coniferous trees, right? So our gymnosperms are our pines, our cedars, our spruces. Now, only 1,000 of them, 1,000 species, still exist. Whereas if we're looking at our angiosperms, our flowering plants, they are the most diverse land plants, and there's over 300,000 species of angiosperms, so many more. And our angiosperms are the younger of the two from an evolutionary standpoint. They evolved around 160 million years ago, didn't really become widespread until a little bit after that. So we are breaking down our plants, right? So we had our seed plants versus our spore plants. And then with our seed plants, we had our gymnosperms versus our angiosperms. And now if we're looking at our angiosperms, we can further divide them into monocotyledons or monocots versus dicotyledons or dicots. Now, a cotyledon is an embryonic seed leaf. So within the seed, there is a structure called a cotyledon, and that is essentially a leaf. So our monocots, as the name suggests, mono being one, have one cotyledon, whereas our dicots have two cotyledons, di meaning two. That's the important distinction. Now, besides just their number of seed leaves, there are a number of other physical traits that can help differentiate monocots versus dicots. Now, they're not always true. Sometimes there's some variation in these. But for the most part, our monocots, if we're looking at their root structure, our monocots have fibrous roots, whereas our dicots tend to have one thick taproot. If we look at the vascular system, we're going to learn in maybe 2 lectures from now about the vascular system of plants. In our monocots, the vascular system is kind of scattered. With our dicots, it forms this nice ring around the stem. If we look at the venation of our leaves, monocots tend to have parallel leaves, whereas dicots are more net-like. And then if we look at the petals of the flower, in monocots, they typically come in 3s or multiples of 3s, whereas in dicots, it's 4 or 5. Multiple, or I guess, I don't know if we see multiples of that. Possibly. So again, some distinctions, some physical distinctions that can help you identify monocots from dicots. However, the real true distinction is that monocots have one cotyledon, one seed leaf, whereas dicots have two cotyledons, two seed leaves. So I kind of talked all about that because what we're leading up to is monocot and dicot germination. And that's what's going to be the rest of this lecture. But when I talk about germination, what I'm talking about is the sprouting of a new plant from a seed or a spore. In this case, we're just looking at seeds, but it would work for our spore-producing plants as well. Now, for germination to occur, there are a number of requirements. The dormancy of the seed must be overcome. We need certain environmental requirements, such as the right temperature, water, air. Sometimes there's some other requirements as well. We're going to get into more of that later on in this course. But what I want you guys to get from this lecture is the pattern of germination. That's what I'm going to be focusing on. So there are 2 main patterns of germination with our angiosperms. There is hypogeal germination, which is below ground. The germination takes place below ground, and that is typical of our monocots. Whereas epigeal germination takes place above ground, and that's typical of our dicots. And I'm going to explain that below ground, above ground in a little bit. So we're going to start and we're going to look at a monocot. And this monocot is going to do hypogeal germination. But we're going to start in the beginning. We're going to look at the seed, right? Because, again, germination is the sprouting of a new plant from a seed. So let's look at a typical monocot seed, and that we're going to look at our corn seed here. Now, I should mention our monocots are our grasses, right? They are our grasses, our cereal grains, some other flower species as well. Orchids are monocots. But if you think of monocots, you're going to tend to think of your grasses, so all of your grains. Now, if we look at the monocot seed, we'll see that at the outside of the seed, there's a seed coat, and that acts to protect the seed. And then there is this large section of the seed that is the endosperm. And the endosperm is really important in monocots because it is the food store for the developing embryo. So the endosperm is essentially starch, which is sugars that are combined together to form larger carbohydrates. But it also contains some protein and oils, fats as well. Now there is a outer layer, the special outer layer of the endosperm, which we refer to as the alurone layer. And that plays an important role during germination. So the cells of the alurone layer, once germination kicks off, they begin to secrete amylase, which is an enzyme, and that amylase breaks down starch into sugar. And it's that sugar that our developing embryo is going to use to essentially grow, right, to power its metabolism and to grow. So that is the alurone layer. Now, if we move to the actual embryo, we'll see there's a number of anatomical parts that you need to be aware of. Again, our monocots. Monocot is short for monocotyledon, and cotyledon is the seed leaf. Now, in our monocots, the cotyledon kind of has two parts. There's the scutellum, which kind of acts to absorb the nutrients that are produced in the endosperm, and it feeds the growing embryo those nutrients. And then there's the coleoptile, which is a sheath-like leaf through which the primary stem is going to grow. And that primary stem is called the plumule. You'll also hear it referred to as the epicotyl. It's the primary shoot. And on the other side, we have the radicle. And the radicle is the primary root, and that grows through a sheath-like structure called the coleorhiza. So that is our typical monocot seed. And if we look at how germination works, again, this is hypogeal germination. And what this means is that those cotyledons, in here, the scutellum and the coleoptile, are going to remain below ground. Now, there are a couple of advantages to this type of germination. Again, remember I said that the majority are monocots are grasses, and if you think of grasses, you think of plants that are grazed upon, right? So this type of germination protects the new plants, the new developing embryos, from grazing. Right? So even if as it's growing it gets grazed upon, there's still this food store in this endosperm in the seed that can allow for new growth to occur. In monocot germination, it is also, because there's this food store, this endosperm, we typically need fewer nutrients initially as it's growing and less sunlight. So these can be found in poor soils or in areas where, like the middle of the forest, where there is poor sunlight. So what is the actual pattern of germination? So what happens, first step in germination is the seed is going to need to take in water. It's called, it imbibes water. That seed takes in water. As it does, that kicks off all this seed metabolism and all these processes. That alurone layer starts secreting amylase, all this other stuff starts happening. And the first structure that is going to begin growing is the radicle. And that's what you'll see first, this radicle emergence. And again, this radicle is the primary root. Now, any roots that branch off from the radicle are called secondary roots, right? So these are roots that are branching off from this main primary root. And that can go further and further. So if you get a root that branches off from the secondary root, that would be a tertiary root, and it goes on and on. But that first primary root is the radicle. And again, that radicle is growing through this coleorhiza, which is this sheath that is covering the radicle and it's acting to protect the radicle as it grows. As the radicle is growing and we begin to see some roots branching off from the radicle, we have the plumule begins to grow, and that's the primary shoot. Now the plumule begins to grow, and it's growing, it's being protected by the coleoptile, and eventually that plumule will break the surface, and as it breaks the surface, we have the development of the first leaves, and our plant begins to further grow. Right? So not super complicated. What we're going to do now is watch a little video of it. Alright, so bam, first structure that is emerging. That is the radicle. And again, that radicle is growing. It's being protected by a sheath called the coleorhiza. Before all this kicked off, that seed needed to imbibe water, took in water, a bunch of metabolic processes started happening. That alurone layer began secreting amylase. The endosperm started breaking down into individual sugars that the embryo could then use to start growing. So we have our radicle is emerging. Our radicle is continuing to grow, and there we start to see the emergence of the plumule. Again, that plumule is the primary shoot that's going to give rise to the main shoot of our plant. That plumule is growing. Oh, and I should have mentioned that plumule grows through that sheath, which is the coleoptile, which is part of the cotyledon. So there it's growing. Oh, and we got fuzzy here, but as it breaks the surface, you see the development of our first leaves right here. Over here in our root system, we start to see the development of secondary roots. And those are going to continue to spread. And we have the development of our leaves, more roots. And there's our corn. All right, so pretty easy, right? Not too complicated. A couple anatomical terms you guys need to know, but the actual process, fairly simple. That is hypogeal germination, typical of our monocots. Now we're going to take a look at dicot germination. Again, dicot, 2 cotyledons, 2 seed leaves. So our dicots are a lot of our vegetable crops, trees, shrubs. They are dicots. Now, if we look at the dicot seed, we'll see it looks a little bit different. You don't see that big endosperm store, right? It's not there. Instead, what takes up most of the room in our dicot seed are these 2 cotyledons here. And these cotyledons contain a little bit of endosperm. So they're going to help power the growth of this embryo as it develops. Also, within our dicot seed, we have, again, our seed coat. We have our radicle here, which is going to be our primary root. And then instead of the plumule, we have what is referred to as a hypocotyl, which is a stem that you'll see exists below these cotyledons. And then we have the epicotyl, which is going to be our plumule primary shoot, and that's above the cotyledons. So let's take a little look at our dicot germination. Now this is, instead of hypogeal, this is epigeal. This is above ground. And really what that above ground means is that our cotyledons are pushed above ground. In our hypogeal germination, the cotyledon, the scutellum, the coleoptile primarily remain below ground, and the plumule gets pushed above ground. Here, it's a little bit different. Now, let's take a look at it first, and then I'll talk a little bit about this other stuff, right? So again, first beginning steps are the same. The seed takes in water, it imbibes water, that kicks off all this metabolism. The first structure to emerge is the radicle. The radicle begins to grow downward, that is the primary root. Secondary roots can branch off from the radicle. The difference here, instead of that plumule growing up through the seed, we have the emergence and elongation of this hypocotyl. And this hypocotyl is the stem below these cotyledons, and it begins to grow, and as it grows, it breaks the surface and it pulls along with it the cotyledons and the remaining embryo. Now these cotyledons will open up and they will act as kind of leaves. They will do some photosynthesis, and as the plant starts photosynthesizing, it starts producing its own food. So these cotyledons will help produce some food in some of the plants, not all of them. Eventually they're going to shrivel up and die, and that is because the first true leaves get produced and they begin taking over the role of photosynthesizing. So with our dicots, a couple of big differences, right? There's no endosperm store, And so because there's no endosperm, there's not a lot of food stored up for the growing embryo. So our growing embryo needs to get up through the soil relatively quickly and into the sunlight because it needs to start producing its own food through photosynthesis. So these are going to grow relatively fast, right? Starting from the beginning of germination, it needs to get this embryo up out of the soil surface for these cotyledons to start producing photosynthesizing and producing food, as well as the first true leaves. So now, let's take a look at that process. All right, there is the emergence of that first root structure, which is called the radicle. You guys got it. So there's our radicle growing down. And there, oh, I'm going to stop it. Okay, as these primary roots are growing off from our main root, our radicle, we start to see the elongation of this structure. And this right here is that hypocotyl. And as you're going to see, it's going to form this crook and it's going to drag up with it the rest of the embryo. This here is the seed coat, as you can imagine. And here you can see the two cotyledons peeking out. So let's continue. So there, that hypocotyl is expanding, it's pushing up through the soil, it kind of forms this characteristic little hook, and it pushes up through, and now it's exposed. And there, let me pause it real quick. Okay, so you see we have our radicle that has continued to grow, that's our primary root, and we have secondary roots branching off. Here is our hypocotyl that has pushed the embryo up through the soil surface. These are their cotyledons, and you can already see the first true leaves poking out that have developed. So our plant now is continuing to grow, and it expands. So this is the hypocotyl right here. Here are our two cotyledons. There's some seed coat still left on. And this structure from the cotyledons upward is the epicotyl, or you could refer to it as the plumule, or as we're going to see later, it's the apical meristem. But that is going to be the primary shoot now of this plant. So now it continues to grow. And again, in some of our dicots, these cotyledons are photosynthesizing. They start photosynthesizing as they're pushed up out of here and providing the plant with some food. But now that we have our first true leaves developed, they are going to be doing a lot of photosynthesis and providing the food for the rest of this growth. And our roots are continuing to expand. And our bean is growing. And let's see how big. This is 3 minutes. I don't think we need to watch all of this because really we're just focused on germination. But it's kind of cool to just see, isn't it? And the music's good. Bum, bum, bum. All right, I think that covers it for now, right? So again, the big differences that you're seeing here is you don't have that endosperm store, and instead of this plumule growing up through the ground, you have this expansion of the hypocotyl that brings along the rest of the embryo and those cotyledons above ground. And that is it. That is the lecture. So again, I want you guys, from this lecture, I want you guys to know that we have our seed plants versus our spore plants. Within our seed plants, we have our gymnosperms, which don't produce flowers, and our angiosperms, which produce flowers. Within our angiosperms, we have monocots, which have one seed leaf, and we have our dicots, which have two seed leaves. Our monocots are our grasses, our dicots are kind of everything else. Our monocots do hypogeal germination, which takes place below ground. And I want you guys to be able to know those structures of the seed and kind of trace how that germination occurs. And our dicots do epigeal germination. Again, know the structures of the seed and be able to trace how this germination occurs. All right, that is it for today. You guys have a great rest of the week.